Hello, my name is Alexandra Davidoff, and in this presentation, I will be discussing antimicrobial resistance and the discovery of novel antimicrobials from soil isolates for therapeutic purposes of creating new antibiotics. Antimicrobial resistance occurs when microorganisms such as bacteria, viruses, fungi, and parasites evolve to resist the drugs that they were once effective in treating infections caused by them. This means that the treatments that we rely on become less effective or even useless over time. AMR is a natural process, but human activities have accelerated its spread. Misuse and overuse of antimicrobial agents contributes to the development of resistance. AMR has become a global health crisis with far-reaching consequences. According to the World Health Organization, AMR is responsible for an estimated 700,000 deaths each year worldwide, and this number could rise to 10 million by 2050 if current trends continue. Infections caused by resistant bacteria are becoming more difficult to treat, leading to prolonged illnesses and higher mortality rates. The WHO has declared AMR one of the greatest threats to global health, food security, and development. One of the major challenges in combating AMR is the lack of new antibiotics being developed. According to the WHO, no new class of antibiotics has been introduced since the 1980s, and fewer than 10 new antibiotics are currently in the pipeline. This lack of new antibiotics is creating a dangerous gap in our ability to treat resistant infections. Given the urgent need for new antibiotics, we turn to soil, one of the largest reservoirs of undiscovered bacteria. Soil contains a diverse range of microorganisms that produce natural compounds to protect themselves from other microbes. By isolating soil bacteria, we aim to discover novel antimicrobial agents that could help combat resistant infections. My 2021 soil isolate that I am using for analysis came from near the UF Beef Teaching Unit, a 90-acre farm with cows. The sample could have likely been influenced by animal manure, agricultural byproducts, and nearby road traffic, leading to a large microbial diversity. As seen in the pictures, we decided to test our isolate for any antibiotic resistant or sensitivity it may have, using a Kirby-Bauer assay. We found that the isolate was resistant to chloramphenicol, novabiocin, oxacillin, penicillin, rifampicin, tetracycline, sulfonamides, and vancomycin. It showed susceptibility to gentamicin and meropenem. Inputting our bacterium's FASTA sequence into the comprehensive antibiotic resistance database, it identified several resistance genes that can be seen in our Kirby-Bauer assay results. Cat86 confers chloramphenicol resistance, VAN-T and VAN-Y create vancomycin resistance, and AID-F and QACJ and QACG encode efflux pumps, which contribute to quinolone and tetracycline resistance. The isolate showed no significant antibiotic production against 18 tested bacterial strains, including E. coli DH5 alpha, Micrococcus luteus SK58, and an E. coli library of various resistance plasmids. Additionally, a modified Hodge test was performed, which showed no presence of carbapenemase production, aligning with the Kirby-Bauer meropenem results. Using anti-SMASH, we identified 24 secondary metabolites in the 2021 soil isolate, producing eicosalides AB, rhizosomide, AB, collocin, rirapentide, all with 100% similarity scores. These compounds exhibit significant antimicrobial activity against pathogens like Streptococcus pneumoniae, Staphylococcus aureus, and Trypanosoma brucei, highlighting their potential for therapeutic use. We also tested the formation of biofilms in our isolate and found that silver nanoparticles reduced biofilm formation by 27.2% after 48 hours. This suggests that silver nanoparticles can inhibit the isolate's biofilm development by inducing oxidative stress, disrupting quorum sensing, and reducing the production of extracellular polymeric substances. Through bioinformatics analysis, the 16S rRNA analysis identified the isolate as Bacillus pulmonis, while the similar genome finder via the BVBRC matches to the Candidatus Canoplastibacterium dezuzai. However, the Candidatus proteobacteria cannot be cultured in the lab. As the results show, we successfully cultured the isolate and performed tests like Kirby-Bauer, which strongly suggest that it is most likely the Bacillus pulmonis species. Bacillus pulmonis is a gram-positive, rod-shaped bacterium that's non-pathogenic and commonly found in soil. Notably, this bacterium is resistant to UV radiation and hydrogen peroxide, which helps it thrive in harsh environments. This resistance typically emerges during periods of environmental stress. Additionally, the isolate has applications in fungicides, where it's used to inhibit the germination of some fungal spores. The isolate shows significant resistance, especially against critical antibiotics like vancomycin, oxacillin, and tetracycline. Also, though it shared similarities with Candidatus Canoplastobacterium desicide, it is very unlikely that it is actually that bacterium. Secondary metabolite analysis uncovered extensive promising compounds that have strong antimicrobial potential. However, the lack of inhibition in our initial antimicrobial assays might actually be due to the testing conditions not aligning with the natural environment of the isolate. Therefore, further testing under more varied conditions is crucial to better understand the full potential of these compounds, which could lead to the discovery of new antibiotics that can combat resistant pathogens. We then collected a new soil sample to isolate, this one taken from a flower bed in the back of the microbiology building, as shown in this image. We tested the soil isolate for antimicrobial production by evaluating its ability to inhibit the growth of DH5-alpha, a gram-negative bacterium, against SK58, a gram-positive bacterium. The isolate showed no inhibitory effect against E. coli DH5-alpha, suggesting that it might not affect gram-negative bacteria, which are generally more resistant due to their outer membranes. However, it did have slight antimicrobial activity against M. luteus SK58, indicating it might have some weak antimicrobial production. We then performed traditional diagnostics, plating the isolate on blood, nutrient, McConkey, and TSA agar plates. On blood agar, the isolate displayed varying hemolytic patterns, including alpha, beta, and gamma hemolysis, suggesting the ability to lyse red blood cells. McConkey agar showed minimal growth, with only a couple colonies on the highest concentration, indicating that the isolate is likely gram-positive, since McConkey selects for gram-negative bacteria. TSA plates produced uniform colonies, but blood agar supported a broader range of growth with a higher CFU per mil counts. Using the MALDI-TOF mass spectrometer, we determined the isolate was Bacillus cereus, a gram-positive toxin-producing bacterium commonly found in soil and food. It causes gastrointestinal illnesses like nausea, vomiting, and diarrhea, and can lead to serious infections in immunocompromised individuals, such as septicemia and endophthalmitis. The bacterium's pathogenicity is linked to its production of harmful toxins like hemolysins and proteases. It can also survive in extreme climates and thrives in diverse conditions. This presence of hemolysins and Bacillus cereus aligns with the observations we saw in our blood agar of alpha, beta, and gamma hemolysis, indicating that these toxins are present in this bacterium we identified. For future directions, in order to discover more antimicrobial properties of our two strains isolated, B. cereus and Bacillus pumilus, we need to investigate other virulence factors to assess the broader health impacts it can have. We can do this by testing the isolate's resistance to more antibiotics and now in various conditions to see how that impacts the resistance patterns. However, most importantly, we need to explore the soil environment further for more bacteria and discover novel antimicrobial properties. We also isolated bacteriophages from wastewater in Gainesville against Serratia marcescens as an alternative to traditional antibiotics. Phage therapy targets specific bacteria, offering a promising solution to antimicrobial resistance, especially against resistant strains like the one that we isolated with Bacillus cereus. Initially, no visible plaques appeared from my water sample, but after using a classmate's successful plaque, I was able to purify and enrich the bacteriophage, leading to visible plaques on TSA plates. The phage titer ranged from 5.9 to the 10th to 1.9 to the 11th plaque-forming units per mil, indicating a high concentration of phages was enriched. The host range test showed that the phage could infect some Serratia marcescens strains, but not others. Additionally, the phage was able to significantly reduce biofilm formation of some Serratia marcescens strains. These results show that the bacteriophage is effective against certain strains and could be a valuable tool for targeting biofilm-associated infections. And thank you so much for listening to my presentation. I have had a lovely semester. And I have really, really, truly enjoyed this class. Thank you so much for all of your help and guidance, and I've learned so much that will be very, very useful in my future career. And these are my references that I used in my presentation. Thank you.